He did his PhD at San Diego State and did his postdoc at University of Hawaii. He currently serves as an adjunct faculty at San Diego State, and his research focuses on how viruses and bacteria affect biogeochemical cycling, energy flow, and community structure in coral reef systems. And he is now a McCurdy visiting scholar at the Marine Lab. We are so thankful for him to be here, also to provide insight onto coral research, which I think is super duper cool. Anyways, thank you, Dr. Roach.
Yeah. You'll have one of these things that works, right? If not,
it's plugged into the main computer.
I was, well, not really to advance my slides, I just wanted to point here, which I can wing it.
Director.
Yeah, so thanks for having me. Thanks for coming to listen. As the title alludes to, I'm going to kind of talk about using omics and imaging technology, and kind of looking at the diversity of coral reef life at a very broad scale. So not just at these like kind of macrological scales, but like kind of looking at the unseen players of the reef, so the bacteria and viruses and small molecules, things like that. So I'll kind of jump right in. Well, let's see. No, it's all good. I think I just was not, didn't want to advance the slide the way I had it. Is that shared on the screen with the other people?
Not yet.
No?
But it's got a 7-minute delay or a 7-second delay. I'm not saying anything.
Okay.
Okay, now there's.
Okay. Yep. So for those of you who've never seen a coral reef, they look something like this. This is one of the actual reefs that a fair amount of the data I'll show you today comes from. This is a reef in Curacao, and this is a 3D model that we made of this reef. Kind of give you an idea of what a coral reef looks like. So when I'm talking about them today. And as most of you probably know, coral reefs are pretty important. They provide for fisheries, they provide for natural products discovery. I think one of my favorite things that they do is over geological time, these ecosystems are actually making islands and island chains and kind of changing the geography of what we see on the map over long time periods. But despite their importance, we kind of see this ecological phase shift occurring all over the world, where we're going from these healthy, pristine reefs shown here on the top left, kind of typified by calcifying organisms like corals and crustose coralline algae, to these more degraded systems shown here on the bottom right, which are mainly characterized by non-calcifying organisms. So you can picture seaweeds, but nasty algae, things like that. There's really two main things that push reefs towards this degraded state. One being local stressors, so things like algal competition, nutrient runoff, overfishing, and then global stressors, so things like global or ocean warming, ocean acidification, things like that. And I'll talk a little bit about both of those in the talk today, and then I'll kind of finish up talking about how we can use what we've learned to push these reefs back into a healthier state. So I'll start off talking about local stressors, and the exact stressor I want to talk about is this direct coral algal competition. And we'll kind of move from that scale into larger scales. And so one of the ways that we think that coral and algae interact with each other on the reef is what we call the DDAM cycle. So DDAM stands for dissolved organic carbon, or DOC, disease, algae, and microbes. And the basic premise is that as algae starts to inhabit the benthos, that algae releases dissolved organic carbons into the water. So you can just picture those as like dissolved sugars, but that's bacteria food essentially. And those dissolved carbons cultivate what we call a copiotrophic bacterial community. So copiotrophs are bacteria that thrive in these really energy-rich environments when you have all this carbon in the water. And those copiotrophs eat up all that carbon, and in doing so, they use up more than their fair share of the oxygen. When they draw the oxygen down, this kills the coral. Less coral on the benthos means more room for algae. More algae means more DOC, more copiotrophic microbes, less oxygen. And you end up with this positive feedback loop that ultimately results in coral mortality and algal dominance of these ecosystems. And of course, this doesn't just happen overnight. This is the long-term cumulative effect of direct coral algal competitions like the one shown here. So we've got a coral on the left, we've got an algae on the right, and a lot of what I'll show today kind of focuses on what we call the interface. So this direct interaction zone between the two organisms. And if you know anything about kind of what I do, I'm not just looking at these organisms as a macroorganism. So I don't just care about the coral and the algae. I'm really interested in what's called the holobionts. So a holobiont, you can picture that as like a meta-organism. So it's not just the coral, it's not just the algae, it's also all of the bacteria, viruses, fungus, small molecules, chemicals, everything that's there. And I'll kind of talk about some ways that we look at all these different components of the holobiont. And so one of the first ways that we study this in the lab is actually what I think is a really kind of elegant experiment. So we took coral and we put it next to algae in a tank, and we separated them by a 0.02 micron filter. So what that means is that the coral and algae don't actually make any direct contact, and they can't exchange bacteria or viruses between the organisms. What they can exchange are things that are dissolved in the water. So things like sugars and nitrogen compounds, things like that. And we had some really striking results. So when we have coral by itself with no algae, we have 100% coral survivorship. Everybody's doing great. When we put algae on the other side of the filter, we have 0% coral survivorship. So that's 100% coral mortality. So you don't need stats to tell that like there's a major effect here. But interestingly, when we add antibiotics to the mix, even with the algae, we alleviate that mortality and restore the coral survivorship back to 100%. So this indicates that there's some sort of role of bacteria in mediating this algal-induced mortality. I think what's really interesting about this experiment is we have algal-induced coral mortality, even though there's no contact between the coral and the algae, and it's microbially mediated, even though there's no exchange of microbes. So it really brought up this question like, well, what the hell is actually causing the coral to die? Because typically we think about like disease and things, you need to exchange the microbes. So to kind of follow up on this, we set up this small scale microcosm experiment. We put coral and algae next to each other in aquarium, and we outfitted these things with a planar oxygen optode, which I'll explain more about in a little bit. We rigged up these custom sampling ports that let us suck mucus and water and whatever else is in that mucus and water, so bacteria, viruses, et cetera, off of the surface of the coral, off of the surface of the algae, and from the interface between the two organisms. And we process those samples for calorimetry, flow cytometry, and epifluorescence microscopy, and I'll touch on those methods here in a second. So what is a planar oxygen optode? So these are plastic sheets that we coat in a chemical cocktail, and those chemicals will differentially fluoresce depending upon the oxygen concentration in the surrounding water column. So we can actually visualize in 2D the oxygen concentration around these organisms. We've actually done this in 3D now using like microscopic beads, so we can get a 3D oxygen dynamic in the water column. And so what we have here is we have coral on the right, algae on the left, we have an oxygen optode behind them. On these optodes, green means more oxygen, red means less oxygen. And you can clearly see that we developed this hypoxic area between the coral and the algae. And this is repeatable time after time. It's more extreme during the night because things are producing less oxygen, but it's there 24 hours of the day. So this is starting to hint that maybe this lack of oxygen is what's actually killing the coral, that it's not an actual microbial exchange, that maybe there's microbial oxygen use is what's killing the coral. We then took samples from the coral, the algae, and the interface, and we put an aliquot of that sample in this machine called an isothermal heat conduction microcalorimeter. Most of you probably aren't familiar with these machines, but this is a machine that allows us to directly measure the thermodynamic power output of a sample. And the way it does that is it holds those samples at a constant temperature, and it measures the energy input or output needed to keep them at that temperature. So this is a way we can directly measure the thermodynamic metabolic output of the microbial communities that we just sucked off our coral and algae. We measure that in energy use per unit time or in watts. And so what we see when we do that, we have the heat output on our vertical axis and the time on our horizontal. You see that at all points in time, we have a higher heat output from those microbial communities associated with the interface. If we take the area under those lines, we can calculate the power output. So that's the energy use per unit time. So you can picture that as the metabolic output of those communities. That's what's shown in the inset here. We have nanowatts per gram. And you see that those bacterial communities associated with the interface have a significantly higher thermodynamic power output than other communities. And I know that's like a little hard to interpret, but so shown here, this is like your kind of standard, what we call a yield to power curve. And you see that there's this explicit trade-off between how fast you do things and how efficiently you do them. And so this FCR, functional control region, is kind of where biology makes its living. And basically you can interpret that higher power output, meaning that these bacteria are releasing more energy as heat. And so they're wasting more of that energy. That's less energy that can be used to do things like build biomass and reproduce. So there are these like fast, sloppy bacterial communities that are developing at the interface. We took some of those samples too and threw them in a flow cytometer. And we used the flow cytometer to basically tell us, these are cytographs that you're looking at. We're basically trying to understand how many autotrophic bacteria are there. So how many bacteria are there that can produce their own food through photosynthesis? And then how many heterotrophic bacteria? So how many bacteria are there that need to eat carbon from something else? And what we see is that in these interface associated microbial communities, we have a significantly higher heterotroph to autotroph ratio. So these communities are getting more and more heterotrophic at the interface. We then also took samples for epifluorescence microscopy, so we can directly count the number of bacteria and viruses. So behind this is a micrograph. And so all of the big sparkly things are bacteria, the little shiny dots are viruses. And when we do that, we see that there's no total difference between the total amount of microbes that are there. So it's not, I guess I'll go ahead and show the next slide. I just did some basic correlation analysis here to see what best explains this increase in metabolic output. So we've got our heat output on the vertical axes on both of these. On the left panel, we have heterotroph to autotroph ratio on the horizontal axis. And you can see that the heterotroph autotroph ratio explains a pretty fair amount of that change in metabolic output that we're seeing from the microbial community, whereas the total amount of cells explains basically none of the variance. So this indicates that perhaps that augmentation in net heterotrophy is what's actually causing the change in this metabolic output of these communities, and ultimately what's responsible for that oxygen drawdown there at the interface. So I think we kind of, nailed things relatively well in this tank experiment, but you never know if that has any relevance to what's actually going on out in nature. So we followed up on this in the field. We went to these 3 island chains. So we went to Papahanaumakuakea, which is a group of islands off of the Hawaiian chain, kind of out in the middle of the Pacific Ocean. We went to an even more remote island chain called the Line Islands in the remote Pacific. And then the Lesser Antilles, Curacao, Aruba, Bonaire, and the Atlantic in the Caribbean. And we found coral algal interactions while we were there. And we suck water. The first thing we do is we suck water off of them, just like we did in the tank experiment. But then we also take small biopsies, and I take a biopsy of the algae, a biopsy of the coral, biopsies of the interface where I get a little bit of everything. So we process those water samples the same way we did in the first experiment. We look at them for cell size and abundance using flow cytometry and microscopy. But then we also use these omics technologies. Are you guys familiar with omics? So omics are any technology that allows you to look at a large class of biomolecules all at once. So you've probably heard of genomics. Genomics lets you look at all of the DNA in a single organism. It looks at all the DNA in a single organism. Metagenomics lets you look at all of the DNA in all of the organisms in a sample. So if I took a sample of your skin, I would sequence I would sequence the bacteria that are there, the fungus that are there, the viruses, literally everything. So when we take a sample of the coral, we sequence the coral, we sequence the algae that live inside the coral cells, we sequence the viruses, the bacteria, the fungus, everything all at once. The metatranscriptomes are all of the RNA in all of the organisms, and the metabolomes are all of the small molecules. So these, you can picture this as these metabolomes as like a biochemical fingerprint that doesn't include protein. We didn't do proteomics. Well, we did, but we didn't include it in the study. And the first thing we did is we looked at these cells that we sucked off the coral and the algae, and we looked at them under a microscope to see how big they were. And so here on the vertical axis, you've got cell volume, and in these, and then you've got your sampling areas across the horizontal. And in these graphs, every dot is an individual bacterial cell. And what you see is, this is a zoom here, so you can kind of see the significance better. But we have way bigger cells at the interface. So at a per cell level, we have much larger cells at the interface than the coral or the algae. I then use this equation here called metabolic theory of ecology. And I won't kind of bother you with the mathematical specifics, but basically what it does is it let us take those cell sizes that I just showed and estimate the metabolic power output. So at the time, we could not bring a calorimeter into the field, even though we've engineered and built our own calorimeter that's pretty close to field deployable at this point. But basically, this was our best way to estimate thermodynamic power output without bringing a calorimeter in the field. And what we see when we do that is that at a per cell level, we have significantly higher power output of these interface bacteria. So this supports our calorimetry data from those tank experiments. And of course, it's not just, you can't only rely on those per cell measurements because the community dynamics is actually, it matters what each cell is doing, but it also matters how many cells are there. So this is just showing that we do indeed have higher total community biomass at the interface. That's shown there in the top panel. And then we have higher predicted community power output. This really does seem to back up what we found in the initial study. So the next thing I did, now that I know that I have increased metabolism, pretty sure at the interface, I went into the genes, I went into those metagenomic data, and I started to look at all of the genes involved in photosynthesis versus all of the genes involved in respiration. So this is basically a way for us to back up that flow cytometry data and see if we have more heterotrophy, but at a genetic level, right? So this is just another layer of kind of proving what we think we see. And you can see that at the interface, we have a significant overrepresentation of genes involved in respiration relative to genes involved in photosynthesis. So we do have increased heterotrophy at the interface, even in situ. And to kind of take that one step further, and I know this gets a little nitty gritty, but I started to ask, okay, if they're all doing, we're increasing the heterotrophy, but what type of heterotrophy, what type of respiration are they doing? So I don't know if any of you have taken biochem. If you have, you've probably seen this in your textbook. This Embden-Meyerhoff-Parnas pathway is like your textbook canonical way to do glycolysis. You go from glucose to pyruvate. However, there's these alternative ways, like this one shown here on the right called the Embden-Deuteroff pathway. There's a few other alternative ways. And I got really into the thermodynamics of it all at this point, and I spent about 3 years of my life going back to first principles and trying to figure out what we would predict the thermodynamic power output of these different pathways to be. We basically derived this equation here that relates the minimum entropy production, so you can picture that as like how wasteful these reactions are, to the thermodynamic path length of these reactions and the number of steps in the reactions. And so since we're going from the same starting place to the same finishing place, the thermodynamic path length is the same for both of these pathways. But you'll notice that this pathway on the right has less steps. So what that means is that pathways like these EMP pathways, and this has been shown empirically now, that these pathways like the EMP pathways are more efficient in the sense that you get more output per unit input. So I get more ATP per unit glucose. You can kind of picture it like that. But these other ones they're faster. So I get more ATP per unit time. But they're faster at the expense of being efficient. So we call these slower, more efficient pathways yield pathways, and these faster, less efficient pathways power pathways. And what we see when we look in that metagenomic data is that we have a significant overrepresentation of genes involved in power pathways like the ED pathway and this pentose phosphate pathway, another pathway we're interested in. But those genes are enriched at the interface, and we have a significant underrepresentation of genes involved in these yield pathways at the interface. So it's not just that these microbial communities are becoming more heterotrophic and doing more respiration, they're actually shifting what type of respiration they're due to this sloppy, really fast way of doing it, which is what allows them to outcompete everything else for the oxygen that's there. So we think that's why they're actually able to draw down the oxygen so low at that in that interface community. And once again, we think that's what's ultimately responsible for the coral dying. So this isn't just true at this like small scale level where the coral and algae are directly interacting. This actually scales up to whole reefs. So what I'm showing here, these green parts of this two-way dendrogram these are different reef sites across a bunch of different islands. The more green those boxes are, that means there's more algae, presumably less coral. The less green they are, the more coral, less algae. And what we see is that in algal-dominated reefs, we have an enrichment in these power pathways and a depletion in these yield pathways. So this scales up to the entire water column. So it's not just something that's happening right there at the coral-algal interface. This scales up to whole reefs and presumably whole ocean systems. So what we kind of learned from this study is that as we have benthic space begin to free up, we get more algae. More algae means more bioavailable DOC. We have some interesting results on that if you're interested about kind of the actual thermodynamic properties of algal carbon, which make it cultivate these heterotrophic communities. And then Once we get increased heterotrophy, we then kind of have this yield to power switch, and those power-driven microbes are able to outcompete coral for resources like carbon and oxygen. And so we've really moved from this DDAM model to what we now call DDAMIT, which is basically the same thing as before, except for now we've involved the interface thermodynamics. And we started to model this. We're using this multi-physics platform to model fluxes and concentrations of all these different things. Dissolved organic carbons, inorganic carbons, nitrogens, viruses and phage, all sorts of stuff. And this is kind of just an example model. In this model, you can see we've got a flow velocity that comes from the left and move to the right. We've just modeled the algae as squares and the coral as a hemisphere. But you can see that we're able to capture this dial flux. So we have, during the day, the oxygen is coming out of these holobionts. During the night, the oxygen is being consumed by them. And you can see that this does indeed, like we capture these hypoxic zones at the coral algal interface. So our model's at least working at some level to kind of give us the cycling and the fluxes of all these different components. To kind of take this further, we're starting to go from these 2D small scale models to starting to build 3D models. And what that means is that we're swimming a shit ton of circles around corals, photographing them, thousands and thousands of photos of a single coral and all the organisms that interact with it. We get these high resolution 3D models of our corals that we're interested in. Once we find a coral that we're into, like this coral in the middle, you can see we've got all the organisms interacting with it. We then come back and we sample it for pretty much everything I could think to sample it for at that point in my PhD. But we take about 100 samples from each coral. We take samples from all the things that the coral's interacting with. We process them for all the ohms and everything we can think of. We're also, I really just love to show these next models. I can't help myself, they're just too sexy. But we're also using UV light at nighttime to excite the green fluorescence protein that naturally occurs in coral and literally making glow-in-the-dark 3D coral models. So they're not just sexy though, they actually also tell us a little something. What we found is that the interface of corals that are winning in the competition against algae, that are currently overgrowing the algae, have a high GFP to red fluorescence ratio, whereas corals that are losing against the algae have a low green fluorescence to red fluorescence ratio. So this is potentially like a kind of quick and dirty way to assess whether a coral is going to win or lose in its competition with algae. But once we make these 3D models, we take all the samples, what we can do then is we can actually go back and map all of these different genes or molecules or RNA, whatever we want to look at, back onto the 3D surface that we've created. We can create these 3D molecular maps. And once you find a molecule that you're interested in, like this one, this is a lipid that we were interested in, you can actually look at its distribution in 3D on the coral surface. And like I said, we took a bunch of different ohms. Each one of them kind of tells its own little sub-story. If you're really interested, you can read the paper. But I think one of the more generalized things that we found in this study was that there's no signature of isolation by distance or spatial autocorrelation. So in most systems, if samples are closer to each other, then they're more related. But we see zero, like none of that at all. And we've repeated it quite a few times. And we don't see any of these signatures of distance really driving the community structure. We see that it's more driven by the ecology. What's next to you? Are you diseased? Things like that. But we did get this paper published, but all three of the reviewers asked the exact same question. They said, what if you did this with a higher sampling resolution or you did this at different scales? Maybe you're just not capturing it because it's an artifact of your methods. So in response to those reviewers, we decided to develop a single polyp multi-omics technique. So this allows us to really sample like the fundamental unit of a coral. We can go in and take a single polyp. We're doing this with some pretty small polyp corals. We're getting, some of these samples are about half the size of a grain of rice. And we're able to get RNA, DNA, metabolomics, proteomics, pretty much everything out of this. And we're doing it in a way that's actually really simple and straightforward. You basically just take these things, dump them into methanol, dry your methanol off, ship your sample wherever you want it to go. But once we developed this kind of single polyp multi-omics technique, we wanted to use it to kind of revisit these questions about the spatial structuring of coral ohms. And I'm going to use metabolomes as my example. So these are small molecules that we're looking at. For the rest of the talk, I'll pretty much only talk about metabolomes, although we've done other work. And so what we did is we went and we got 19 colonies from across a reef. We then took the most eastern branch and most western branch and a branch right in the middle from each colony. And then from each branch, we take three polyps that are touching each other about 1 centimeter from the base. We take a polyp at the very tip and then we bisect that distance twice. So So we have a nice fractal design. It's a self-repeating spatial design, which covers at least 5 scales of magnitude from like millimeters to kilometers to really address that question that the reviewers asked. Like, you can't get a smaller resolution. You can't really go much more scales unless you wanted to go across island chains. So, this really gets at like those questions of resolution and scale. The first thing that we saw is that we have a major signal of genotype. So the genotype of the coral matters. It's not really mind-blowing, but it shows that we have nice, clean data. It does tell us that, okay, if you're going to sample corals, you need to sample the same coral each time. If you're going to do time series, things like that. But I think what was more interesting is that we have this really strong signature of sampling area within a branch. So these are principal component analyses, or actually this is a canonical plot, kind of same difference. But this is, I don't know if you've ever seen these things, but these are basically ways to take, we get thousands and thousands of metabolites that we're looking at. So we can kind of compress all those thousands of variables into 2 dimensions and see how things fall out in this space that we've created. And you see that basically polyps 1, 2, and 3, they're down here at the base, they're touching each other, form one little group. Polyps 4 and 5 in the middle form another group, and then the tips really, really different than everything else. But I think what was most interesting is this percent misclassification or misclassified up there. We used a neural net to basically tell me how well it could distinguish between polyps. And that neural net is actually able to distinguish between polyps that are touching each other with about 96%. It's actually, well, yeah, 93% overall, but just in those polyps that are touching each other, we can get it to like 96%, which is almost horrifying actually. Because that means that if I sample like one polyp up towards the tip, I am going to significantly influence my results. But it really tells us something about how spatially structured a coral holobiont is. And we see that it's, once again, it's not really distance that matters, it's how close you are to the tip or the base, not how far you are away from each other. So kind of interesting results there. And kind of what we're doing moving forward on all this is we're starting to try to like develop these multi-scale dynamical models. So going from a single polyp, looking at the fluxes and fluxes of different compounds in and out of those polyps, then scaling this up to whole colonies and then whole reef systems. We're actually at this point right now where we're using this exact 3D model as an input surface, and then we're modeling hypoxic zones, zones of high nitrogen enrichment. We can actually see where hypoxia is most likely to pop up on the reef. Then we go check for it and ground proof it and test it and see if our models are actually working correctly. So really starting to scale up from these like simple 2D models to 3D systems. I'm gonna shift gears a smidge and go from these local stressors and now kind of talk about these global stressors. In particular, I'm gonna talk about temperature stress. So many of you may have heard of coral bleaching, which is what you see here on the right. And basically coral bleaching, so I guess I'll take a step back. Corals are colonial animals that have an endosymbiotic algae that actually lives inside their cells. These algae are oftentimes responsible for 90% or more of the coral's energy needs. And during times of stress, the corals can puke those little algae out. The reason they do that is to like get rid of a lot of free radicals that they produce during stress times. And they turn white, hence the name bleaching. And if they stay bleached for long enough, the corals can die because they've lost their energy source for too long. And so what we did is we went out in the field during a bleaching event in Hawaii in 2015. And we found these coral colonies that prior to this bleaching event, we thought these were all one colony. So you can see them here. This colony on the right resisted the bleaching. The colony on the left did not bleach during thermal stress. The colony on the right did bleach during thermal stress. And like I said, if a month before this, you would have never been able to tell they were 2 separate corals. But what we did is during that bleaching event, we went out and tagged these corals, and then we waited to see which corals recovered. And we came back 4 years later during a time when there was no temperature stress. So everything's healthy and happy. And we resample these corals. That's really important because up until this study, basically everything that had ever been done to look at coral bleaching was looking at coral during active bleaching. And it's like, well, I don't need metabolomics to tell me that a bleached coral is different than a non-bleached coral. Like I can have an undergrad just look at them and be like, that one's white, that one's not. So it's important to note that during the sampling of this study, everything is visibly healthy. There's no differences in the coral visibly. And what we saw is, so these are PCOAs again, and we see that once again, we have a really strong signature of genotype that's shown in the colors of the circles, which is nice because that supports our previous data. But we also see, which is shown by the shading of the circle, that we have a really strong signature of whether a coral bleached 5 years previously or not. So we have this like signature of historical bleaching. This is not at all what we were expecting to find. And it actually, our neural nets were able to classify it based on about 10 molecules. I could use about 10 molecules and classify these with 100% accuracy. So really, really strong. Like so much so that I didn't believe my own data. So I went back out and I found corals that we didn't know anything about. I took samples from them. We used our models to predict which ones of those corals would bleach and wouldn't. We exposed them to heat and we were able to predict their bleaching phenotype with 100% accuracy. So I think we have a really strong model of what's, kind of deriving this bleaching, at least from a metabolomics perspective. And to try to figure out what those molecules were, we used a random forest algorithm. It's kind of like a really basic learning algorithm. And what I'm showing here is called a variable importance plot. As the name implies, it literally just lists variables in order of importance. So it takes all of those thousands of different metabolites and it ranks them in order of how important they are for determining if a coral bleaches or not. The very first molecule is a beta-ene lipid. It's actually beta-ene lipid 16-0. I'll talk more about it in a moment. But you'll see that there's lots of orange bars towards the left-hand side of the graph. So there's a lot of these beta-ene lipids that are popping up as important variables in determining bleaching. And so we started to look at these, this beta-ene lipid was our most important one. And we started to look at its difference between the different phenotypes. And what you see is that non-bleached corals have a whole lot more of this specific biomarker, this DGCC 16-0 lipid. But even more generally, what we really see is just that non-bleached corals, the corals that don't bleach, have a higher saturation state of all their lipids, which kind of makes sense if you're into biochemistry or physics or thermo, that you can think of it like butter, right? It's like more saturated. It doesn't melt and come apart as easy as something that's less saturated, easily degrades due to heat. So it makes a little bit of sense that non-saturated things would have higher thermal or more saturated things would have higher thermal tolerance. To take this a little further, we went and separated the host and the symbiont. So we take coral only and algal only, and we look at just the metabolites from the symbiont or the algae and just the metabolites from the host, and then the metabolites that they share. And I think this makes it even cooler. We see that we still have a really strong signature of whether a coral will bleach just based on just the algal metabolites and also just the coral metabolites. I think what's most interesting maybe on these graphs is that we actually have a signature of the coral host genotype and the algal metabolome, which hints that maybe there's like biological, or like immunological crosstalk between the two members of the symbiosis. They're kind of both deciding who gets to be there. So kind of interesting stuff. But the big takeaway is that we still see this signature of whether a coral will bleach or not under heat stress in both members of the symbiosis. So we then went back and we took these same corals that had previously bleached and corals that hadn't bleached. And we were actually thinking that if we expose them to really high heat stress, that we might make the corals that had never bleached start to look more like the bleached corals, right? You like try to bleach them, maybe they'll look more like bleached corals. And we actually saw exactly the opposite. So the clear circles, once again, are corals that do bleach. The gray circles are corals that don't bleach. And you see that when we expose them to heat stress, they actually become even more different than each other. So this tells us that there's like an active response in those corals that don't bleach to turn on all these different metabolic pathways to try to resist the bleaching. And so we see that, like, if you look at the holobiont as a whole, we see that. But if we look at each component of the holobiont, they're actually all responding differently. So we see that the symbiont compounds actually all kind of respond in the same direction. The host compounds kind of respond in the same direction. And the microbial compounds are just all over the place. So this tells us that, the host and the algae, the coral and the algae kind of have very directed ways to handle heat stress. And the microbiome is a little more stochastic, which kind of makes sense because it's probably not the same between every coral. So kind of interesting stuff, I guess. And then to really push this study kind of as hard as we could, we went back to those corals 5 years after they had bleached, and we waited for them to spawn. And it's a really cool phenomena. These corals spawn on the darkest night of the year at exactly 8:20. Well, I guess the 3 darkest nights of the year, but you can set your watch, kind of float around in the water, and also you see all these orange and pink balls float up through the water. So we collected egg and sperm. We then take those egg and sperm and we selectively breed the bleached parents with bleached parents and non-bleached parents with non-bleached parents. So we selectively breed for those different bleaching phenotypes. So what I'm showing here, this is just kind of an idiot check that like, okay, our data is clean. We see that adults form a really nice group. We see that eggs form a really nice group. Embryos and larvae and juveniles are kind of right in between eggs and adults, which is nice. And then sperm are really, really different than everything. Like I said, this is not surprising. This just tells us that we had good, clean data. We were able to pick up some signal. We have difference in life history stages. But what, I guess, this is what we were looking for, so we weren't so surprised, but what it's, you never know if you're actually gonna find what you want, is that we do indeed maintain this imprinting of those past bleaching events across generations. So we have a phenotypic difference in the adults, the eggs, the embryos, the larvae, the juveniles, and the sperm. And we see that that's particularly enhanced in the embryo, larvae, and juvenile. So that means that, maybe during these early life history stages, the corals are actually really turning on these different metabolic pathways to make sure that they can withstand stressors during these like more critical life stages. Once again, we were able to show that that's true in both host-only compounds and algae-only compounds. So we have the data played really nice. We did the same random forest algorithm to try to look at which molecules are most important at predicting bleaching phenotype in each life history stage. So these are variable importance plots again. The blue bars mean that those things were significantly enriched in non-bleached corals. The yellow bars mean they were significantly enriched in bleached corals. Luckily, this doesn't always happen, but we found the exact same molecule in our adults to be the most important thing. So that it's really nice when the data does that we keep finding repeatable trends. But importantly, we also found these other beta-ene, these DGCC lipids, basically as being important things all across different life history stages. So we really went back down this DGCC lipid hole even further. So this is just a graph showing that our favorite molecule, DGCC 16-0, is enriched in non-bleached corals at all life history stages, except for sperm. They don't actually have it at all, probably because sperm don't have algae. So this is an algal compound, we think. We're 99% sure. And then this next graph is just too pretty not to show, but this is just showing that as the corals develop, that at 15 hours post-fertilization, they're actually all pretty similar in their concentration of these biomarkers. But as they develop from 15 hours to 120 hours post-fertilization, you see this difference between the phenotypes just gradually increase. So once again, kind of hinting that maybe these like early developmental stages are really important for turning on these pathways to resist stress. And then the thing that actually makes this study important and cool is that we're not just able to increase those biomarkers through selective breeding, we're actually able to increase their thermal tolerance through selective breeding. So we have control corals. Those are corals that we just scoop sperm and eggs off the top of the water. They're just wild type. And we have our corals that were selectively bred to withstand temperature stress. You see that you have significantly higher survivorship of those selectively bred corals when we stress them with temperature. So this is a nice potential restoration technique. What this means is that I can go out and take a tiny little grain of rice size sample from my coral, look for different metabolites in it, find those biomarkers to know that my corals aren't going to bleach or that they'll withstand temperature stress. I can selectively breed them and then use those selectively bred corals for restoration efforts. So rather than just like kind of all willy-nilly restoring a reef with random corals, this is a more climate-wise restoration strategy that ensures higher success in future climate scenarios. We started all this work in Hawaii, but we've now set up these bleaching libraries and climate-wise restoration sites in Curacao, in the Atlantic Ocean, and in Sri Lanka in the Indian Ocean. So we're covering all three ocean basins where tropical corals exist and kind of setting up these different things around the world. And I think this is really important because this allows us to see if this is like a thing that only happens in one species of coral, or does this happen in lots of species of coral? And prior to this effort, I think there was like 11 species of coral that we had metabolomes on. And I think now we have 100 and some different species that we've collected. So really trying to dig into like what role coral diversity plays in these trends. So now I'll kind of talk about maybe what's the more interesting part is how we use this information to restore a reef or conserve a reef. So I've already kind of touched on these what we call assisted evolution techniques. So we're doing things like selective breeding, we're stress conditioning things, acclimatizing corals, we're modifying the algal symbionts through selective breeding of the algal symbiont, conditioning of the symbionts, even some genetic modification. We're also doing things like assisted gene flow. But then there's this other suite of restoration strategies that I call personalized medicine on an ecosystem scale. So this is kind of like stealing ideas from human medicine and applying them to ecological systems. So we're doing things like probiotic treatments, mucus transplants, phage therapies, and gene therapies. Does everyone know what phage therapy is? Cool. So phage, it's short for bacteriophage. They are viruses that only infect bacteria. So what we can do with the phage is we can use them to kill off very specific strains of bacteria, much like you would an antibiotic, but they're way more specific. So I can even go in and just turn off one specific gene. So maybe I go in and knock out those ED pathways that I was talking about earlier, right? But this is a way that I can really specifically engineer my microbiome to do the things that I want it to and not do the things that I don't want it to. And so I'm gonna talk mostly about the probiotic treatments, but pick my brain about the other ones if you're interested. This is something I've done in collaboration with Raquel Peixoto's lab. And so basically what we do is we take a coral, we crush it up and isolate bacteria from it. We then screen those bacteria for things that we think are putatively beneficial. So things like nitrogen fixation, antimicrobial properties, antagonism against pathogens, reactive oxygen scavenging, things like that. Once we find bacteria that meet those criteria, we then sequence them to make sure there's no pathogens in them. Then we recombine them back into these probiotic consortia. So I want to make it really clear that we're taking bacteria from the corals and then giving them back to the coral. But we're giving them back in a very different concentration, right? Very different relative abundances. And we've done this for 3 different species of coral: Pacillipora acuta, Montipora capitata, Parietes compressa. And what I'm showing next is basically taking the probiotics from all 3 of those and giving them all to just a single coral. So we gave them all back to, I think, Montipora. And so what I've got here on the vertical axis is FVFM. So this is a measure of quantum yield. It's a measure of photosynthetic efficiency, but you can picture that as like how bleached is the coral. And you see that in ambient conditions before we heat stress them, all our different groups are the same. But once we put them under heat stress, shown here on the right, you see that our control group that we just squirted filtered seawater on does significantly worse than all of our probiotic treatment groups. So that means that we can elicit a thermoprotective effect by giving them probiotics. So we can make corals withstand heat and not bleach by giving them probiotic consortia. But of course, there's still a lot of kind of big unanswered questions. So how do we apply these strategies at whole reef scales? Are there off-target effects? Are they gonna fuck up some other organisms? And then how long do these effects last? And so we need a way to kind of assess the feasibility and risk of these approaches. And one of the places that we're starting to do that is this place here, the Biosphere 2. You've never heard of it? It's a very cool structure out in the middle of the Arizona desert. It was originally created by a bunch of hippies in the late 1980s. They found a billionaire to bankroll it. No one that built this thing had a PhD. The only requirement for working on it was that you had communal living experience. And like I said, they were able to build this in about 1.5 years. It's the world's largest set of enclosed ecosystems. It's about 4 acres under glass. It goes about 12 stories up and 2 stories underground. I've lived here for about 3 months at a time. And then in the 90s, they actually enclosed 8 humans inside this thing for 2 years and 20 minutes. They were able to produce all their own food, scrub all their own CO2, get all their own oxygen made by plants. It was a hermetically sealed system that I think is about as tight as the space station. And they were able to live in there for 2 years. There's some drama and scandal. You should check it out if you're interested. But inside the biosphere, they've got, I think, 6 different ecosystems. So we've got a mangrove ecosystem that has all the different genera of mangroves in the world. That connects to an ocean system, which I'll talk more about. We have a rainforest system, which is largely Amazonian in nature, with like a really big waterfall in the middle of it. There's an orchard that grows fruit trees. There's a coastal fog desert. There's a savanna. The Landscape Evolution Observatory is now an experimental lab where they look at landscape evolution, but it was originally where they grew all their food and had their chickens and goats and things when they lived in there. And then the habitat is where they would have lived when they're in there. And one of the coolest things I think are these lungs. They're giant geodesic domes, and they actually allow the biosphere to breathe. So because the desert has such extreme temperatures, and PV equals nRT, right? I'm sure you guys know that one, ideal gas law, right? when it gets cold, you know, everything shrinks. And so you run the risk of this thing exploding or imploding when it's completely sealed with those temperature changes. So inside the lungs, they have the world's largest single rubber gasket that holds up a 20,000 ton piece of concrete and steel. And during the day, those pieces of steel rise up, and during the night, it pushes the air back out through the bronchioles underground into the biosphere and actually allows it to breathe. They have some of the craziest acoustics I've ever experienced. They are really fun to go in there and rage out at night. Yeah, it's very cool. Lots of echoing and weird. The sound actually rolls around the walls and stuff, so you can like whisper and talk to people on the other side of the room. Very cool. And of course, the main attraction as far as we're concerned is the Biosphere 2 ocean. So this is the world's largest experimental ocean system. It's about 7 meters deep, about 1 million gallons, and about 850 square meters of benthos to play with. And I got hired for my first postdoc to try to engineer a coral reef inside this thing. I was not super successful, but we're still trying to do it. And currently what we're trying to do, because this had a lot of hysteresis and was actually a pretty degraded system, we've been pushing this system back, and now we're starting to implement these coral reef restoration technologies that we call coral reef arcs. So arcs are these geodesic structures. They float mid-water. So they try to restore reefs off of the benthos. And so that gets us away from the hypoxia that's driven by the algae that I kind of discussed earlier. And so this is an artist's representation for a book that I've been writing for the last few years of what an arc could look like. And the idea is that we seed these things with coral, and they eventually turn into these kind of like floating zoos that we could, and I've done this now, we can unhook them from the bottom and move them to a new reef and let them seed a reef to kind of as a conservation effort. But these aren't just sketches in a book. These are actually real existing structures that we've implemented. We've put them in Puerto Rico, we put them in Curacao, and we're starting them in Madagascar now. And you can see that we seed them with what we call arms. So these are coral that we've collected, and we put them on there. You can see that over time the corals start to grow, they start to develop a community. You'll notice in the next little bit, they actually start to develop quite a big pelagic community. Fish start swimming around them. They develop their own food webs. I think I would say they are pretty successful. We have control sites where we explant coral in a more traditional manner onto the benthos. Those control sites, we have about 20 to 40% of our coral that live through the 3-year trial period. And on the arcs, we have about 80% of our coral that lives. So an improvement. And like I said, as far as changing the kind of food web, this is my last visit to the arcs in Curacao, and you can see that the like wall of fish behind me is so thick that it like literally blocks the sunlight out. So these things are creating like really intense ecosystems of their own. And the basic idea is that we would take these things called autonomous reef monitoring systems. They're PVC, like little, picture them like reef condos. And they've been used throughout the years to collect reef biodiversity. And basically the idea is that we would use these arms to seed the arcs, and doing some kind of back of the envelope calculations, we estimate that if we put out 10,000 arms for 3 years across 10 different reef regions, that we could collect about 85% of all reef biodiversity and then translocate that to an arc. Once you have them on the arcs, you can then, like I said, pick those arcs up, move them wherever you want them, put them out and allow them to seed different reefs, and use that as a way to kind of propagate and preserve coral biodiversity. I wanted to real quickly just shout out this book that we're writing. It's called Phages, and it's basically kind of zooming in on the beneficial role that viruses have in ecological health and in human health, and kind of talking about taking this more like view of the holobiont for human health and also in ecological restoration, that you really can't just do it by just moving macroorganisms around. You got to figure out the root cause and involve the microbes, and particularly the viruses. So I'll kind of end with talking about like what I'm actually doing at Duke Marine Labs. So I'm originally from Moorhead, North Carolina. So basically right there. And I got a visiting scholar position. And what I said I'm going to do is understand how farmed oysters and oyster reefs affect surrounding viral and microbial communities. Look at the role of viruses and microbes in mediating oysters' response to stressors, and then eventually develop probiotics and phage therapies for shellfish aquaculture. And just as a kind of real quick, we have done some cool experiments with oysters at the aqua farm, and we're actually finding that oysters selectively eat autotrophic things, and it's not driven by size. So oysters are tasting different bacteria, and they're selectively eating the autotrophs. And it's significantly changing the heterotroph to autotroph ratio around the reefs and presumably significantly changing the oxygen and carbon dynamics of the ecosystem. They also look like they're actually really affecting the predator-prey dynamics of virus and bacteria. So when we add oysters to the system, we get Lodka Volterra dynamics in our predator, in our bacteria and viral communities. So kind of interesting stuff. I didn't put it in here. We literally got the data back like a week ago. But some cool stuff going on there, I think. With that, I'd like to thank everybody who helped make all of this stuff happen, especially all the students. All the people that gave me money for it. It wouldn't be possible without them. And I'll take any questions or heckling or whatnot.
So like you described how from one generation to the next, they like passed on this resilience to coral bleaching. Were those like I guess like initial chemicals you measured expressed because of coral bleaching events, or were they inherently in the?
Yeah, that's a great question. So when we first found all this, we really thought that it's a real chicken or the egg thing, right? And we really thought that it was like we were calling it metabolomic imprinting. We thought that like they were changed because they bleached. But now the more that we do it, we actually think it's the other way around. They don't bleach because they have these chemicals. And I think especially those heat stress experiments kind of point to that like, oh, and when we try to heat them up, they actually become even more different than the ones that bleach, right? So I really think it's that they don't bleach because they have those chemicals, not that it's like imprinted on them after they bleached, but it's still a little bit TBD.
On these arcs that you start putting in, have you tested or have you seen like any type of difference in the interface interaction as compared to like the bottom? Like, does coral win out at a greater percentage?
So it's a little bit hard to say. We do see a difference like anecdotally, but I think part of the difference is that there's no established algal communities on the arcs for the most part, right? So it's like the algae's kind of like trying to play catch up, whereas on the benthos, there are already massive algal communities. So we don't really see these like kind of D-dam dynamics on the arc. Like we don't see like what we call algal feeding, where you're like feeding the microbial communities algal carbons and they're getting all out of control. So yes, they're different, and it's unclear if it's like just a time thing, like if over time enough algae will accumulate. But I really think that that's actually just not the case. I think that they will be actually different. And I think in part because there's so much more flow in the midwater. So if you have laminar flow and you have a surface, the water or fluid hits that surface and creates what's called a diffusive boundary layer. So that means that what's happening in that boundary layer is all governed by diffusive dynamics. And that doesn't really happen in the midwater, right? You've got flow from all these different directions. You don't create these diffusive boundary layers the same way you do. And so you don't have the same problems with like, oh, like you don't get hypoxia because you just get oxygen brought in from the water column, right? Whereas on the bottom, you get hypoxia because you get this little boundary layer that forms and then you can't diffuse in oxygen fast enough. Does that make sense?
Yeah.
So I think that they are actually different, but don't really know.
Have you ever thought about like cold stress, like impacts on these corals? Like, do they kind of like get like, does coral have the same features like resisting the heat stress, kind of tolerate cold stress?
Yeah, so that's a really good question. There have been people who have done some of that. You can elicit bleaching through cold stress, specifically in like temperate corals. And so basically right now at Duke, which I didn't plan on doing this, but over the last month, I've basically started taking oculina corals, like temperate corals that we have here in North Carolina. And we're gonna try to do an experiment where we induce bleaching with heat stress, induce bleaching with cold stress. And then we also induce bleaching through like vibriosis. So we give them vibrios, which are a specific bacteria that we know can be involved in bleaching. And so basically try to see what the differences are. And like, I should mention that those oculina actually exist sometimes naturally with no symbionts, even without stress.
And Australia too, coral.
Yeah, exactly. So, right, so we have those, and so we can use the ones that are unstressed but aposymbiotic versus the ones that are aposymbiotic because they've been stressed, to kind of tease apart like what is a signature of symbiosis versus what is like a generalized stress response versus what is a heat specific stress response. bacterial specific stress response, cold specific. So trying to start to tease that apart in a model system at Duke, or at the marine labs. Yeah.
Have you seen any like adverse effects of adding the probiotics? And could that mess with the health, like the healthy biotic community that helps the coral?
Yes, so we did, like, this is all anecdotal at this point, like, we did get lesions on some of the probiotic treated corals that when we resequenced them, they had Vibrios in them. We don't give them Vibrios specifically, so we know that we didn't give them those Vibrios, but we created some sort of niche space that allowed them to bloom. And Vibrios are really interesting. It's like, sometimes they're good, sometimes they're bad, sometimes they're completely innocuous, like doing nothing. There looks like a lot of temperature dependence pathogenicity in Vibrios. So I can say that we did see some probiotic treated corals exhibit these like tissue lesions, and that it looked associated with Vibrio potentially, but there's no like statistics to support what I'm saying. That's just like vibishly after doing the experiment, we noticed some of that. But by and large, most of the metrics we were interested in performed better with the probiotics than without them. And it's a little unclear if that's actually because they're like being probiotics or if the corals are like just eating the microbes or something. Like we don't really know the mechanism, but they are at least correlated.
Does it seem to be kind of luck which corals have the parts that make them heat resistant, or like, is there anything you can use to predict that?
Yeah, so the answer is no, it's not luck. It actually really looks linked to what symbionts you have. But then that goes a step further, that what symbionts you have really looks linked to what immune molecules you have. So it's like corals have specific immune modulator molecules that are like picking out their symbiont type. And then what symbiont type you have really determines if you resist bleaching or not. So I didn't show this because it gets a little messy, but basically like about 90% of our corals that don't bleach have these cladocopium symbionts they have durasdinium symbionts, and about 90% of the corals that do bleach have these cladocopium symbionts. So it really looks driven by what type of algae you have in you. But like I said, that's not just like random. It's not like I just randomly uptake some algae. It looks like the corals are kind of specifically selecting those algae to be in their cells or not. That makes sense? Can I answer your question?
You guys decide where to put the arcs when you integrate them into like the wild type coral communities?
I always say it's when one of two biologists agrees. Like everyone's got their own metric that they're like, this is how you determine it. One of the things that I've been doing lately, which gets real nerdy real quick. So we've basically I use those Lion Islands that I was talking about. They're like some of the most pristine reefs in the world. They're super isolated, way far away from humans. They're thriving. They do great after bleaching events and things. They recover quickly. So we've been looking at the different ohms from them. So metagenomes, metabolomes, things like that from the water column on those reefs. And then we go out and find degraded reefs that we want to restore. And we run those same ohms. And then we use what's called a Kullback-Leibler divergence. It's a relative entropy. So basically we compare the distributions of the things that are, let's say, bacteria in the water at our pristine reef versus at our degraded sites. And we can calculate this entropic distance between the different sites. So I can say that like, oh, these sites are microbiome-wise closer to the pristine sites, even though they're degraded. So that gives me an idea that like, oh, maybe those sites are more amenable to restoration. But a little bit further, that Kullback divergence metric is actually asymmetric. So like going forwards is not the same as going backwards. So we can also look for sites where the backwards divergence is smaller than the forward divergence. And that lets us know that maybe those would be the easiest to restore based on the omics data that we have. So we've been like kind of slowly hacking that method together to try to publish it and make people actually believe it. Because right now it's more like a harebrained talk I gave at a math symposium, but we're working our way through it. I think it's something that I think it will be usable, because the thing is that everyone wants to restore the sites that look the worst, but oftentimes those are the sites that if you restore them, like everything's just going to die again. So you need to find a way to target sites that are really degraded, but that have some sort of markers that indicate that, oh, we could actually restore them if we tried. So I don't know if that answered your question well, but that's one of the things we're trying to do. I have, if anyone's interested, I have a paper on cobaltly divergence you can go look at on how that works. But
Have you found that there is like an ecological, look, how are the arcs affecting the ecosystems at in those water columns? And like how, is there an adverse effect or like, is there some sort of cascade of like, okay, this is a new food source at a different point in the water column?
yeah, so we've done like your kind of like basic workups of like, are there more fish after the ark's been there for 2 years? Is there more, we separate our fish and like, okay, are there more pescivores? Are there more high level predators? We think of that being a hallmark of a healthy ecosystem. We also have really looked at like the virus and microbial dynamics and I'll say by and large, when we look around the arcs, they have all the indicators that they are becoming a more healthy system, that there's not like kind of off target effects or whatever you want to call them. What's unclear though, so like for the microbial side of things, we can kind of say like, okay, they're shifting it into a healthy microbial state. That's good. We don't see any way that that's really like driven by something off target that's actually could be bad. But on the macrobial side of things, it's a little bit like, okay, so there's a bunch more fish there. But presumably those fish weren't all born there like 2 years ago when we put the arcs in. So is it just accumulating fish from outside? And if that's the case, is it now letting people just be like, oh, well, I can just go fish around the arc now. I don't even have to go look, and like maybe they're removing more fish from the fisheries because of that. So that's actually something we're like currently looking into, trying to do like isotope tracing and things like that to try to figure out like, okay, did these fish originate here or did they come from here? Are they just aggregating? What are the kind of like downstream effects of that through fishing pressures and things like that? But currently, no idea.
Are you still planning to keep going with the oyster studies?
Yeah. Yeah, I got a buddy from Harvard who's like he's all in about the oysters. He and I went to grad school together and we're homies, and he like kind of independently decided oysters would also be cool. Both of us have this like dream of trying to 3D print reefs. And so we played around with coral stem cells for a while, trying to figure out if you could like print with coral stem cells to 3D print coral. And we both kind of gave up on that, but we think we have an idea on how to 3D print an oyster reef. So that's how we kind of both ended up on oysters. It was like, okay, I think we can 3D print an oyster reef and we can use that as a proof of concept on how you would 3D print a coral reef. So yeah, still following up on oysters, very interested in their like microbial and chemical ecology of farmed and natural oysters.
So do you think like those temperate corals like Alkalina or Australias can like the same mechanism like as what is in like the tropical corals, like maybe on cold bleaching, especially because they like bleaching is not like a big deal to their survival. Actually, because they kind of like strategically like use the bleaching as like a way like to survive the winter. But yeah, do you think they can reflect what's happening in the tropical coral, or we still need like studies on the tropical coral, especially like those maybe living in the higher latitude areas of the tropics, like South Australia or.
I mean, I think that there's like, there's like broad similarities, and we'll find out because we're like, I should have all this data back really soon to kind of answer that question. So I think at like a kind of high level things like saturation state, things like that, those things may hold true. I kind of doubt that those specific molecules will be the same molecules, because if I've learned anything, it's like a coral is not always a coral. Like there's really big differences across species and across, genera of corals. But they are, so I think high level, yes, but very like more zoomed into like specific molecules and specific pathways. I kind of think those things probably won't be generalizable between temperate and tropical corals. But there's only one way to find out. So we're doing that.
You mentioned before about the importance of like the hollow biome in the environment. How does would the idea of like 3D printing kind of intersect with the hollow biome? Because 3D printing is obviously not like a biological, like it's not alive.
Oh no, I mean 3D bioprinting.
Oh, really?
Literally,
Oh, wow.
literally printing a reef. Like not, so like this guy, Daniel Wangsprat, has kind of taken it as far as you can on corals, or as far as anyone has on corals so far. So like we've been able to 3D print calcium carbonate matrices for a while, which is like basically a coral skeleton. And he's been able to lace those calcium carbonate structures with zooxanthellae. So he can lace them with the algae that the corals would need, hoping that, then when you put corals on there, they've already got the algae that they need. But we're really trying to take it further and like literally 3D bioprint like coral stem cells into a coral. I think like, so you can, they've 3D printed livers and things like that, right? So I think that I think we're getting there. I really want to bioprint a whole arc out of coral. That's like where I'm at right now with trying to think about that. Because we kept coming up with like, what material should we build them out of? And I was like, we'll just build them out of coral. Like, that fixes that problem. So yeah, no, and actually ideas of incorporating microbes and you know, protists and things like that and viruses that would be beneficial to help the printing actually work. I think that would be the idea, and probably is like a multi-community member problem where we're gonna have to print bacteria and coral cells and stuff all at once to really make it work. Printing live shit.
So are there just like universal coral microbes that transcend like your different sites in like Pacific, Atlantic, Indian Ocean?
Yeah, that's a great question. So microbes, not so much. So actually, a friend of mine, Eric Hester, back in like 2013, something like that, did this paper on the core coral microbiome. And he was basically asking like, are there bacterial strains? And he was trying to do it at a strain level, you know, get really strict, that are like in every coral. And basically at the strain level, they weren't able to find a single strain that was in like 3 different species of coral. So I was like, okay, I'm gonna do the same thing in the metabolome. And like I said, I think they had 3 genera of coral. We're at like 67 genera of coral. And we find that like so we find ubiquitous molecules first off, like there's probably 50 different molecules that are in every single coral I've ever collected, which is actually crazy compared to the microbiome stuff. Like I thought we were gonna have none, because that's what happens in the microbes, but we have a lot. And it actually turns out that probably about 80% of the molecules are in 80% of the samples or more. So they're really ubiquitous. And that's actually like currently like a big project that I'm working on, is just like, what is the biodiversity of coral molecules? How much of that biodiversity do we estimate gets lost during bleaching events? If corals go extinct, you know, if species go extinct, how much biodiversity do we lose? Molecular biodiversity do we lose? And how much microbial biodiversity do we lose? And then also just what makes a coral metabolomically. And now because we had so many molecules that were found in every sample, we basically have designed this like outgroup sampling scheme where I'm just like, I started with scleractinias, which are like those tropical corals that I'm working on mostly, and like working my way through the tree of life. So we take like their sister taxa and then we slowly work away and then find other basal metazoans and all the way up to humans. So now we've got a data set that includes from humans to corals, and looking at what are those common molecules. But it kind of makes sense that like if you're you know, that the molecules that you have are more alike than your bacteria, because you collect your bacteria kind of stochastically, either from your mom or from the environment. Whereas like what makes a cell? It's like everybody's got phosphocholine, you know, everyone needs a lipid bilayer and things like that. So it kind of makes sense why